thought you already did.
Okay, so let's talk about the heart. We'll complete that and make it good. So, heart, lungs, heart, cardiovascular. Okay, so the heart pumps, as it says, about 4,000 gallons of blood every day. How do we have that much blood? How much blood do we have?
No,
5 liters. Give or take about just a little bit over a gallon. We just pump the same blood multiple times. So we couple of things. So for those who were in lab, it will be revision lecture. You heard of it previously. But we have to remind ourselves about right side of the heart, left side of the heart. Right side receives oxygen poor blood from the rest of our body. Left side receives the oxygen-rich blood, pumps it to the rest of the body. Right? And we have 4 chambers. 2 are called atria. They are also the receiving chambers, which means blood first arrives to the heart in those chambers. Then it goes to the ventricles, which are also called the pumping chambers, because they're going to pump the blood out of the heart. Where does the right ventricle pump the blood to? To the lungs, while the left ventricle to the body, right? So we'll talk about a circuit. Circuit, you can think of starting point one spot, you're coming back to it. Here, it doesn't have to be the same part of the heart, but it's returning back to the heart. Okay? The first one, the pulmonary circuit, that involves the lungs. So we have oxygen poor lungs coming to the right side again. Remember, whenever we're looking at image, as a general rule, it's looking at someone else's looking at you. So this is the right side, this is the left side. So that's going to start here, receive blood to the right atrium, right ventricle, and from here it's a pulmonary circuit going to the lungs and then back to the heart. Okay? While systemic circuit that supplies the rest of our body will technically start from the left ventricle, goes through the aorta to the rest of our body, and comes back to the right atrium. Okay? Those are the 2 circuits. So, next thing we also need to remind ourselves, is also a conversation in the next chapter, is blood vessels, arteries and veins. One of the classification of them is not by the oxygen-rich nature of the blood they're carrying, but by which direction they're taking the blood. Okay? Arteries take blood away from the heart, veins bring blood towards the heart. Okay? So, away from the heart will be arteries, veins towards the heart. And then capillaries are the smallest of blood vessels where gas exchange takes place, or any exchange takes place. What kind of cells are they? Make up the walls of our capillaries.
Single squamous epithelium.
Yeah, so it's simple squamous epithelium, just single cell epithelial. Right, so thin wall, which allows for diffusion to take place very easily. Okay, so the heart is simply the size of our fists. Each of our own fists, right, our own hearts, and it's sitting in the middle of our thoracic cavity between the lungs. What's that middle space called? Mediastinum, and it's in the inferior part of the mediastinum. The left ventricle is bigger, so it does go more towards the left side. Okay. So roughly starts at the second rib, goes up to the 5th rib, the costal rib, is where we're going to find the tip of the heart, the apex of the heart. Okay. What else did we talk about? Yeah, in terms of the heart, we think of the heart as an upside down pyramid. So the apex of the heart is towards the bottom, and then the base, which we didn't talk about in lab, is where the blood vessel will come. That's the base of it, which is move towards the superior side of the heart itself. Okay? Next, we're going to talk about pericardium. Pericardium is a serous membrane, 2 layers, just like our pleura, just like the third serous membrane we learned. So it's pleura, pericardium, and the not epicardium. So it's pleura, which lungs, yeah. So pericardium around the heart. Other one. Peritoneum, right? Where is the peritoneum covering? If this more specifically, abdominal. That's okay. So this is the abdomen. We do have 2 layers. The innermost layer is called the visceral layer. Going back to tissues lecture. So visceral layer here, 2. Here this is going to be called visceral pericardium. Actually on the wall of the heart. In terms of the people who had lab today, I'm going to change that portion of that lab to more written because we're going to have a lot of written questions. So I'm going to ask what are the layers for the significance of this step. So I'll go through it here. One of them will be here. So pericardium is one the way you had on lab. That's going to be the serous membrane surrounding the heart. That's right. And then who recalls what the names of the walls of the heart was? Anyone remember the list of? So I'll come back to it. Actually, forgot to come back to it. So we have 3 layers of the heart. Endocardium. And it's going to be myocardium. Epicardium. Cardium. To be precise, this is actually the visceral pericardium. Okay, and then we'll have a little space. I have another image I'll show you in a second. And then we'll have the parietal pericardium. Parietal layer of pericardium. This will be pericardial cavity. I'm just going to use a different color. Pericardium. And here will be the lumen of our heart. The cavity of the heart will be the endocardium will be the closest to us. The atria or the ventricle. So for pericardium, we'll say it as it's an outer covering of the heart that holds the pericardial fluid, right? That's really within this pericardial cavity. The pericardial fluid acts as a friction absorbing, right? It's like our brake fluid. Brake fluid doesn't really cause the car to stop, help the car to stop. It's the brake pads that do, but brake fluid just allows the smooth movement. Right? So the pericardium will allow the smooth movement of the heart wall in relation to the pericardium. That's nice motion that produces friction. So 3 layers of the heart. So it's endocardium, the innermost, which is basically the same as our endothelium, which is the innermost lining of our blood vessels. What are the innermost lining of the blood vessels? What tissues are? It's also simple squamous epithelium. Okay? Still simple squamous epithelium here as well. Okay? You might have heard of endocarditis. What's that? Inflammation. The worst part is the endocardium or the worst part of illness has to do with where else it's located. It's not just the heart wall, even the heart valves are lined by endocardium, so endocarditis really affects the valves, and that's when we have more symptoms, valves not closing properly, or actually will actually kind of rupture, because the swelling initially will kind of block, and then later they can even heal, they can start to shoot. The next layer, which is really the thickest part of the heart, is the myocardium. What kind of cells do we anticipate we'll find there? Muscle, and specifically what type of muscle? Cardiac muscle, right? So here, endocardium, the innermost layer, simple squamous epithelium. Then it's myocardium, which has the which made up cardiac muscle cells. That's the thickest of them. Then it's the epicardium, which is also the visceral pericardium. Right? That's by dense irregular. Outer is dense irregular. Here it's areolar tissue. And then we have a space, which is the pericardial cavity. Fluid fills space with a pericardial fluid, and then with the parietal layer. In terms of layers of the heart, we always talk about the pericardium as well. Even though the wall of the heart ends with epicardium. Okay. Moving on, we're talking about the four chambers, right? So right side receives oxygen poor blood, it always arrives at the right atrium. So right atrium will receive blood from the inferior vena cava and the superior vena cava. Where do they bring blood from? Yeah, specifically which one where? Yep, inferior below the heart, superior above the heart. Right? And then we do have another blood vessel which supplies the heart muscle, also we'll talk about that. It's called the pulmonary sinus, which will also then blood will move from the right atrium to the right ventricle and then out to the lungs. Right? By the way, in images and in models, sometimes even in specimens we actually have to inject them, but we use some color coordination to show something. So we use blue for some reason and red for something. What was the reasons for these two colors? Yeah. So really blood is not blue. Okay? Blue, it's always red. It's just bright red when it's oxygen rich. It's a darker red when it's oxygen poor. Hence, so coloring that way. They've chosen to do it red and blue generally. Okay? So again, it's not mean about artery and veins, it's just talking about what type of blood is carrying. Okay? So here, it's oxygen poor blood, hence they've colored it blue, superior vena cava, inferior vena cava. So it'll go through the right ventricle and then into the pulmonary trunk and the pulmonary artery into the heart, into the lungs. Then it comes back as oxygen-rich blood using pulmonary veins and will enter the left ventricle. So another way to also, again, I spoke about this in lab, but for others that have already known this, the best way to distinguish the two sides is looking at the wall thickness of the right versus left ventricle. Which one's bigger? Left. Why? Pumping the rest of the body is pumping at a higher pressure. The rest of the body is at a very high pressure. It needs to build. That's why the muscles are bigger and stronger. Whereas the right ventricle, all it has to do is pump it to the lungs. So left atrium will receive oxygen-rich blood from the lungs, pass it to the left ventricle. That will then supply the rest of the body, systemic circulation. First going into the airway. In terms of movement, atria is going to contract. I do have a slide talking about some how the cardiac cycle works. So we want the atria to contract first before the ventricles. Why? It seems obvious, but we want to think about it. Force the blood into, so why not contract at the same time, ventricles? Yeah, so it wants to wait till it fills up, then it contracts and then it gets sent out. And then we'll also talk about the valves, which will prevent back. So we don't want all 4 chambers contracting at the same time. What happens is the atria contract first, both atria will contract, then both ventricles contract. So we have some grooves as well we'll talk about, but we won't be able to see on any specimen or on a model because usually a blood vessel is in it. Okay? But there are some indentations in them, so we just need to be aware. The coronary sulcus basically goes between the 2 atria and the ventricles of a transverse plane. There are 2 more sulcuses in front, back, between the 2 ventricles. So between in the front, it's anterior, interventricular. What does interventricular mean? Between, right? So inter means between, so it's between the 2 ventricles. Did we look at sulcus in our bones? We did in our brain. What was a sulcus? Central sulcus. What was that? Where was that?
Frontal parietal lobes.
It separated the frontal and parietal lobes. What other sulcus did we learn? The very obvious one too. Lateral sulcus that separated the temporal, into the frontal, a little bit to the parietal. Right. When we talked about the other one, it was not as obvious, but what was the other one we talked about? Parietal occipital sulcus, which basically separates the parietal lobe from the occipital lobe. Right? So sulcus is an indentation and groove, right? Revising what we talked about, what's on either side of the central sulcus? Structurally first and then I'll go to function. Pre-central gyrus, where is that? And post-central gyrus. Is which one? Yep, so pre-1 is in front of the central sulcus, so it's actually in the frontal lobe, posterior. So gyrus is just the ridge, right? We do have many more of those lobes. So here too we have sulcus. But in there we have blood vessels. So generally we do not see the sulcus when we look at any model or any specimen. Again, please remember the heart is like an upside down pyramid. So the base of the heart is actually at the top where all the blood vessels enter the heart. While the apex, where the tip, is at the bottom of the heart. Here portion is the apex of the heart. So let's talk about the blood vessels. We mentioned to the right atrium, it's going to be superior vena cava and inferior vena cava. They bring oxygen poor blood, superior from above the heart, which means arms are included, of course the head, and then below the heart, abdomen and legs will be from the inferior vena cava. We also have another vessel that opens, it's called the coronary sinus. This receives oxygen poor blood from the muscles of the heart itself. Okay? So this will be the ones that are supplying the heart muscles. They will bring it back. We'll talk about this. It's called the coronary arteries. When we go way supplying it, it's called cardiac veins, and then they'll drain into the coronary sinus. I don't know if I mentioned when we talked about the superior sagittal sinus, I mentioned what a sinus was in terms of blood. How is it different? Why didn't they just call this coronary vein?
Arteries take the blood away from the heart, veins bring them back, and they call it the trunk because it's words like that is.
No, the sinus.
Oh, I'm sorry. I misunderstood the question.
Is it because it's still, instead of our sinuses is still with air, not just with blood, but it's the oxygen of the blood?
Yeah, so it's thinner wall, not like a regular vein, which does have muscle. This one does not have muscle. So that's still carrying blood. So these are called coronary sinus. When we talk about superior sagittal sinus, which will drain the CSF into the blood. So this is our then another structure is fossa ovalis. Okay? This is on both sides of the interatrial septum. What would interatrial septum be? Between atria, right? Between the atria, the septum between. So this is now in all of us we should have is fossa ovalis, which is a little indentation in the wall. It was foramen ovale, okay, which basically connected while we were in our mother's womb, because blood. So where does gas exchange take place in fetus? Not in the umbilical cord exactly.
Placenta?
In the placenta, right? So placenta is attached to what?
Uterus.
The uterus, okay? And then how is the placenta connected to the fetus? That's the umbilical cord, right? So that's bringing the blood vessels. So it's all fetal blood, by the way. It's not maternal blood that's in the umbilical. It's all fetal blood. In the placenta is where we have two, one on each side, fetal blood on the fetal side, maternal blood on the maternal side. So blood enters through the umbilical cord. Where does it enter?
Her belly button.
Yeah, that's our umbilicus, right? Does anyone know where it goes after it enters here?
The liver.
It does. It goes into the liver, right? Once it goes into the liver, it's going to come into the inferior vena cava. Okay? This is oxygen-rich blood. Okay, it's coming now into the venous system. So, in a fetus, is the lungs functioning the way yours and my lungs are functioning? No, right? So we don't need to send all of that blood to the lungs. So some of that blood, once it arrives at the right atrium, will go to the left atrium and then join the systemic circulation. Okay? We actually have another one too, but that's what remains of it. So if we have a hole in the heart, number one is this one. This is the reason why it did not close. It takes a couple of weeks after we're born. The first breath we take is already closing, but it completes closure. A couple of weeks. It can remain a little bit longer, but people get breathless, right? We can also have septal defect in the ventricles too. Okay, so hole in the heart doesn't always mean this one, but this is the most common because we had this at some time. Everybody had this at some time. So it was an opening that will just blood flow. It says between, really from the right atrium to the right to the left atrium. Moves blood because it's arriving. Oxygen-rich blood from the placenta goes through the liver, goes through the inferior vena cava, and then you basically want to shunt the lungs. We don't want it to go to the lungs. We want it to go into the systemic circulation. So let's talk about the blood supply from our right ventricle, right? So from the right ventricle is oxygen-rich blood. It will leave the heart, oxygen-rich blood, oxygen-poor blood, right ventricle. So it needs to be oxygenated, so it's going to go to the lungs. It leaves its structure. The blood vessel, the first one, we call it pulmonary trunk. For our course, we want to refer to it as pulmonary artery. It's fine, technically of the trunk. When it splits, this one becomes arteries. That will supply the pulmonary circuit, supplies the lungs. We get oxygen 4, CO2. What's the process by which we exchange that? Like diffusion. Sorry, some of these are probably, hopefully. You've heard of some, but I want to make sure we're talking about all of them. Some may not have heard of, some are okay. So, when it comes back, it comes to the left atrium via the pulmonary veins. And in lab, we'll learn, actually it doesn't come from the front, it actually comes from the kind of the posterior side of the left atrium. We have 4 of them. They don't join like the artery leaves as one trunk, 2 pulmonary arteries to each lung, and then they keep splitting. But here they don't join back. They just come as 4 blood vessels to to the left atrium, to either side. And then it goes to the left ventricle, and then it can enter the aorta for the systemic circulation. Coronary arteries, what do they supply?
So to the heart.
The muscles of the heart. They actually come out the base of the heart. Okay. Most of our organs are supplied better when we are at when the left ventricle's in contraction. Here it's actually not being filled because what's at the base of the aorta? Especially bad people. Aortic valve, right? What is the aortic valve? What's the aortic valve doing when the left ventricle is contracting? Open, right? When it opens, it actually closes off the coronary artery entrance. Okay? In lab, next time pay attention to one of the lab models, you can actually see it right next to the aortic valve. So when they open, they actually close the opening to the coronary arteries. So coronary arteries only fill up during diastole aspect. How many coronary arteries do we initially have? This one I'm hoping we will. Let's have 2 coronary arteries. Left and right. Easy question. Which side is the left one going? Left side of the heart. It supplies all of the left side plus the interventricular segment. Right? The right side, the right coronary artery basically supplies the atria and the ventricle. And we'll talk about branches because you may hear of your patients, they will talk about blockage of one of those 4 branches. Let's talk about the valves in the heart. We have 4 valves. They all prevent backflow, right? We want the blood to go in one direction. If they try to come back, the valve should close, prevent them from coming back. Okay? So we have 2 valves between atria and ventricle. So when the atria contracts, they'll be open, they will let the blood pass through. But when the ventricles contract, we want these valves to be closed. We don't want the blood coming back into the atria. We want them going either into the pulmonary trunk or into the aorta. Okay. Now blood is left. We don't want it coming back to the ventricles. So basically, it's before the ventricles and after the ventricles. Right? So the AV valves. Please recognize that the tricuspid valve, that's the name that's used more often between the right atrium and the right ventricle. So sometimes they may refer to as right atrioventricular valve. The tricuspid is the one you can hear more often. Okay. On the left side, bicuspid, that one has a second name. I expect us to remember both names because it's very likely we'll be talking about that same valve in the 2 names. Okay, you're going to hear mitral valve a lot, you're going to hear bicuspid valve. Okay, so that one is called the mitral or bicuspid. Cusp is just flaps. So this one just has 2 flaps. Tricuspid has 3 flaps. Okay, on the right side, left side, bicuspid and mitral valve. We also have at the base of the pulmonary artery trunk and the aorta. These are semilunar valves that look like moons. They will prevent blood coming back to the ventricles. Okay, at the base of the pulmonary trunk and the aortic trunk. Any questions? Okay. So let's talk about some structures that hold these valves in place or help the valves function. Okay. So we do have separate muscles that moves the valves, opens and closes them. Okay. The muscles are called papillary muscles. Okay, so here there's one shown here. Okay, it's finger-like muscles that jut out from the wall. Okay, and then we have muscles, tendons, right? We have these tendons that are attached to the valve. These tendons are called chordae tendineae because they're string-like, and you're going to see this. People who had lab today saw it. People where Monday's lab will look at on Monday. You'll see the bull's heart. You'll see the nice stringy looking chordae tendineae. So if I had a heart attack, I killed off some of these muscle cells. I killed off the papillary muscle, what do you think I might be suffering with in the future? Yeah, the valve will not be able to close properly. Okay, it's going to be blocked with leaky valves. Okay. So this is important when this muscle contracts, left ventricle contracts, going to pull this, making sure the blood will flow nice and smooth during when atria contract, will actually contract this, and then the valves will open, and then during ventricular contraction, we want the valves to close. Which, if I had a heart attack which affected this, it will not close properly, so blood will start going back to the atria. Which quite happens quite often. It's one of the complications of a myocardial infarction. They have valvular insufficiency if it affects the papillary muscle. So papillary muscle is a muscle. The chordae tendineae is the string-like structure connecting to the back. Okay. Let's talk about heart sounds. You might have heard we have two sounds that we hear. To define it as lub-dub, we need to recognize it's not the contraction of the heart. It's not, what else have I seen? It's not the opening on valve, it's closing of valves, full stop. Okay? The closing causes the sound. So it's the closure of AV valves, the atrioventricular valves, the tricuspid bicuspid valve, which causes the first heart sound, and it's the closure of the semilunar valves that causes the second heart sound. Okay? These are normal heart sounds. Okay? So first heart sound, the lung part, if you will, is the bicuspid, tricuspid closure. Does not make a noise when we open. We should not make a noise when our muscles contract. It's the valve when we close. Okay? Rebound effect. If we hear a murmur, that means there is some the flow is turbulent, or it's not smooth, or it's not laminar flow, it's sinusitis, right? So it can be because if I have stenosis of the valve, that can also make it difficult for blood flow. If I talk about the prolapse or insufficiency, prolapse just means it's going back in the opposite direction, right? The valves kind of going tented like. That leads to insufficiency. Sometimes they're used interchangeably. Really, they are talking one structural change, one is a physiological change. Right? So insufficient basically means it's not preventing that from going back. All those generally cause aortic stem common valvular prolapse and insufficiency. But otherwise, we generally hear two heart sounds. So let's talk about those blood supply first. Okay, the two arteries are left coronary artery and right coronary artery. As I mentioned, left coronary artery basically supplies all of the left atrium ventricle as well as the interventricular septum. Right coronary artery supplies a little less. Okay, it's just the right atrium and the right. The two branches, let me actually draw them out. People seem to remember it a little better. So left, write it up. The coronary artery. We have two branches. That's called the circumflex artery. The other one has two names. I'll put both down. As you a lot of times hear for anterior interventricular artery, or it's also described as left anterior descending. So some of the angiograms sometimes use a three-letter word. These two are the ones most significantly plots, right? The left side. So this usually the right LCX. This is LAD. So they'll say 80% block of LAD, 99% block of LCX. Usually That will be the final diagnosis, but in the description they'll give the proper names. But a lot of times they use this acronym for that. Right? This is where it's worst outcome because it's supplying a larger part, including our left ventricle. Whereas the right coronary arteries also has two branches. Is the marginal artery, the posterior interventricular. So, base of the aorta, so blood vessel coming off the left ventricle. Actually, does a interesting, if you wish, or different. Actually, comes towards the right and then comes out. Okay, it's going to come out. Then the curve goes down. Where it comes out is where the two branches are. Left coronary goes to the left side. Right coronary goes to the right side. Okay, so here is still the left ventricle. That's where it's going to come out from the left ventricle, the aorta. Okay. At the base of the aorta, we have the right and left coronary arteries. The left one, pretty much the short one, pretty much branches out straight away. One goes around, that's the circumflex artery. Yeah. And then the one in front, anterior interventricular artery, which sits in the anterior interventricular sulcus. This is why you don't really literally see it, because this artery is either made, model put down, created, that's the right word, or in a specimen that blood vessels hiding that sulcus. Okay. While the right, it does division a little bit differently, if you wish. Right coronary longer, and then we'll have the marginal artery here, and then it goes around, and then we have a posterior interventricular artery. They've shown it light color to show the posterior side, darker color to show the anterior side of this image. Okay? So you'll see the posterior interventricular artery at the back. We also have, which is good, I guess, we have what's called anastomosis. Anastomosis basically is connection between arteries. So you're going to see, so this one's a circumflex, okay? It's going to meet that right coronary artery at the back. That's the anastomosis. Even our posterior interventricular artery will meet the anterior interventricular artery. That's another anastomosis here. So this is why we're not too worried if the right side blocks, because the left side does supply a little bit of the right side. You can go to the rest, if you will, distal part of the blockage and supply some of that muscle. Okay? Whereas, and before the block, it's anywhere. Usually, we don't have a lot of symptoms. Sometimes we do, but we do not. Whereas the left side is where we generally have more symptoms, more complications. Left branches will be the left anterior descending, and circumflex anterior descending is also called the anterior interventricular. Right side is going to be the marginal and posterior interventricular. These will supply the muscles. So, in terms of veins, we do have multiple trying to collect different. So, three main ones will collect. Small vein, small cardiac vein, great cardiac, and middle cardiac. But the interesting thing is they all generally drain to this common coronary sinus. And the coronary sinus generally, okay, there are few people that the great cardiac vein directly opens up the right atrium, but generally all three will drain. This is at the back in lab. For those who had lab today, it may be called the blue, big, large blood vessel on the posterior side. For others, we will see on Monday. That will collect, and that opens up into the right atrium. Any questions? So let's talk about these two imbalances, right? So we hopefully understand what myocardial infarction is. What is it? Can we explain a little bit? Why does it happen? What is happening? That's the bigger key question. Lack of oxygen to the heart, so hypoxia to cardiac muscle cells. What's happening? Muscles dying. That's going to be the difference between this and another disorder which have very similar symptoms. Okay? It's angina pectoris. So in myocardial infarctions, the cells die. Okay, that's going to be key. And it's usually a blockage that's caused this. Okay? Whereas angina pectoris, similar symptoms are described: chest pains, sweating, nausea, vomiting. Sometimes they have referred pain to the shoulder and jaw. They have the same sense of impeding dew, which is a common symptom that myocardial infarctions patients do express. But here it's short and it's because of spasm and goes away. So usually when we consider as a timeline, usually less than an hour, usually 10, 15, 20 minutes is the usual. But the risk factor is when you keep doing that, eventually or possibly they can develop a myocardial infarction. So angina pectoris patients will be treated as a kind of a pre-MI patient. So they're given similar medications to an MI patient. While here it's already cells are dying, right? And we want to get that blockage sorted out as soon as possible. Any questions? How is this different from cardiac arrest? We have an EMT, so you should be able to answer. Please let me make sure we can answer that. Is it the same as myocardial infarction?
No.
What is cardiac arrest?
When heart stops beating.
Yeah, it stops beating. That's usually electrical imbalance that causes the heart to stop beating. Here, the cells are dying. Good. So let's talk about those cardiac muscle cells. Okay. Striated, what does that mean? Stripes, yeah, colored stripes, so dark bands and light bands. So these are also striated. Are they voluntary or involuntary muscles? Involuntary. Good. How many nuclei per cell?
One, sometimes two.
It's one, but sometimes two. Very good. What else we got? What about the shape and size of the cells? Short, yeah. In terms of shape, you also want to think about branch. These are the only ones that do branch. Okay? And then they have a special connection between the cells. That's an intercalated discs. They have two functions, right? So if you think about, I'm not going to show a branch one again with my markers, but if this was the connection, this could be the intercalated disc. Okay? They prevent, because the heart muscle contracts, relaxes, they must hold these cells together. Okay? That's the desmoid source. They also have what's called gap junctions. We mentioned this in our cell bio chapters, chapter 2 or 3, I forget. What is their function? Gap junctions. So for skeletal muscles, each muscle cell must be innervated. For cardiac muscles, we do not innervate every single cell. Right? So once a cell is innervated, right, action potential forms. What's the first ion that moves in regular muscle? Here, it's actually different. Anyway, let me rephrase that. The first one's the same, the second one's different. What is the ions that move during a regular action potential? We talked about neurons and skeletal muscles having similar numbers and similar pattern. Which one first? It's the sodium moving in, right? Why does sodium move in? We talk about deprotein. Why does it? Why? Make it less. Why does it move in? Where do we have more sodium? Outside. Gate, which way is going to move? into the cell, right? Now let's answer those questions. Why does it depolarize? Why does it become less negative?
Because so in terms of positive charge.
It's positively charged. When the positive charge comes in, it becomes inside normal resting impression. It's negative, it becomes more positive, right? So there, we talked about how kind of the picture of continuous conduction, continuous propagation. I mentioned how one area will not prevent, will actually let sodium move to the next area, causing an action threshold to be reached action. That's kind of like what's going on here, but this is not. Parts of the same cell, but a couple of different mixed cells, right? So it starts, depolarization starts in this cell, some of those sodium ions move here, through those gap junctions, they will cause depolarization of this cell. Okay. This makes sure that we don't have to innervate every single cell. This also ensures that the whole, it's actually both the aqueous, separate out from the ventricles, we'll talk about that, the whole of the chamber will contract as well. Because otherwise you'll have contraction here, contraction there, and so on. It's going to be disordered. So this ensures it spreads throughout. This is through our intercalated disc. It's the gap junction that helps us to achieve that. Where are cardiac muscles found? In the heart, now more specifically, which layer of the heart? Myocardium. Anywhere else? Blood vessels? Okay. So, now let me introduce you a slightly variation. So we talked about it's mostly the contractile cells, right? So they're going to be innervated, they're going to transmit this action potential, and then they'll contract. Okay. This is the bulk of them. But we do have some cells which are non-contractile. They are also called pacemaker cells. Okay. They can auto-generate their own action potential. They don't need a stimulus from our nervous system to start the depolarization of action potential. Okay. They spontaneously depolarize, okay, by themselves. This is what pacemakers, might have said, heard of artificial pacemakers, permanent pacemakers, that takes over the task of these pacemaker cells. Okay. So these pacemaker cells they're not involved in the contraction process. They are more involved in spreading the impulses, not requiring nervous system. If they're beating, heart's beating, how many times a second? How many times a minute? 60 times. We don't have to have the brain telling them 60 times a minute to do that. Okay, it does it without. Okay, and this is part of what's called the intrinsic conductive system. This is like a network, okay, of cells and collections of cells which can self-depolarize, and when one spot does, it transmits to the next one down. Okay, we'll talk about that in a bit. I think we'll break yeah, let's take a break and talk about it. That's the newish topic.